So I'll post it, yeah, probably tomorrow before class, I would guess, but it'll be there definitely before the practical. So one of the big things is growth is not an observation. So outside of like the staff 1 templates where you're only really looking for growth, that's not your observation. You want to say the media changed color or there was gas produced or something like that, but just saying the organism grew for all but I think 1 test is not sufficient. So make sure you have actual observations. For your interpretations, you want to geek out what does this observation mean? And be detailed. So I'll explain some examples a little bit down the line, but I do want full interpretations to get full credit. If you give a partial interpretation, I'll give partial credit, but you're going to lose points if you don't say the full interpretation. So for the first couple, I don't think I have an example of that, but down the line, it'll be pretty obvious what's a full and what's a partial interpretation. So for catalase and oxidase, you'll be expected to know these on the test. You want to know what are the reagents that are used and what are these assays testing for. So does anyone remember the reagents for either catalase or oxidase? What about oxidase? It's kind of a trick question. It's as easy as it should be. So it's Kovacs oxidase reagent. So, and what are they testing for? So what are catalase and oxidase testing for? There I'm not really looking for catalase and oxidase, that's fairly obvious. But what's the purpose of the enzyme? So what do catalase and oxidase do? What purpose do they serve for the cell? So if you think about the actual enzymatic reaction for catalase, does anyone remember what that is? And that should be a big clue as to what the enzymes do. Okay, so catalase converts hydrogen peroxide to water and oxygen. So where does hydrogen peroxide come into play with the cells? Oxidative stress. So catalase and oxidase are both related to respiration via oxygen, and so that's what the purpose of the enzymes is. And for what type of enzymes are they? I'm referring to intracellular or extracellular. So for both catalase and oxidase, they're extracellular enzymes. So there's the broken down version. And as I said, for catalase, know the specific enzyme activity. So I told you that one. You can look up and see what the oxidase enzyme activity is on your own. So this is fairly similar to what you saw in the midterm practical, and you'll be seeing a few of these on the final practical as well. So this is just looking at the media results. So you have a catalase and 2 oxidase tests. So you're going to be required to make the observations and the interpretations for all 3 of these. So does anybody have an observation and the following interpretation for any of these media? Or start off, which one's catalase and which one's oxidase? It's the easiest question.
Bubbles, catalase.
Okay, so what would your observation for catalase be?
Bubbles.
Bubbles.
No bubbles.
Okay, good. And what about oxidase A?
Purple.
Yeah, there's some sort of purple thing formed. And what about B?
No purple.
No purple, exactly. So that's where knowing what the test is and what the possible reactions are comes into play. So if you didn't know what the possible reactions for oxidase were and you only had part B, then you wouldn't necessarily know what to say, right? So it's important to know what they are so that you can know what a positive and negative result is. And if you only see one, to know that that is a result and what it means. So what about interpretations? How about for catalase? What do the bubbles mean? Catalase positive, exactly. And for oxidase, what's the purple and what's the clear? Right, purple's positive and the clear is oxidase negative. So for this one, the observations and interpretations are fairly straightforward. There's not a whole lot to remember. Down the line, some of them are a little bit more complicated. The next media that we'll be looking at is the OF tubes. So what are the OF tubes testing for? Right, so oxidative respiration versus fermentative energy generation pathways. And something that's important to remember is the importance of mineral oil. So why do we add mineral oil to one tube and not add it to the other tube? Right, so the tube with mineral oil is an anaerobic condition, and the tube without is an aerobic condition. And the indicator used for the OF tubes is bromthionyl blue, so that's something that you could also be tested on. So here's some media results for the OF tubes. So the observations for A, B, and C. Now for one like this, when you have two tubes under a single observation, you want to make sure that you do write about both. So for example, I'll just do A. You can write both tubes are yellow, that's fine. I don't need you to say anaerobic tube is yellow and aerobic tube is yellow. But you want to make sure that you include both in the result because both are important in your final interpretation. So what about for B and C? The observations are straightforward. I won't make you guys go through that. It'll be tedious. But what about the interpretations? So for A, what would your interpretation of that be? Right. So when both the anaerobic and aerobic tube turn yellow, that means that fermentation is taking place. What about for B? Right, oxidative respiration. So something that I will note for B is that on this example, the entire tube turned yellow, and that's perfectly fine. But especially I think for a lot of people's unknowns, only the top of the tube would turn yellow, and that's fine too. Since it is aerobic respiration, you would expect that at least the top is going to turn yellow because that's where they have access to the most oxygen. But if the whole tube turns, that's perfectly fine too. And what about for number C? What does two green tubes represent?
Non-saccharylitic.
Non-saccharylitic. Okay?
What did you say the indicator was again?
Bromthionyl blue. Okay, so the MVIC test is another one that you'll be expected to know. So the purpose of this test is multifold, so that it's obviously not a single test. This is the series that's used, but what is the use for the MVIC series?